everyone. Today I'm going to tell you about how science has brought us the first treatments that can slow the progression of the most common cause of dementia, and why this is only the beginning. So first, I imagine you're all very familiar with the concept of dementia, and that's because it is very common. The best estimates are half of us will be directly affected in our lifetimes by dementia, either by developing one of the diseases that cause dementia, or by caring for someone living with dementia. And this huge prevalence comes at a cost. The 55 million people worldwide living with some type of dementia cost our societies around $800 billion or euros per year to care for them. But there is hope in science, which is what we're going to talk about today. But before we delve into the brain and how brain changes cause the symptoms of dementia that we're all too familiar with, which is that loss of memory and thinking ability and ultimately death, first I want to take you back in time. So I want to tell you a little bit about the history of dementia and dementia research. And I'm going to start in ancient Egypt, about 4,000 years ago, 2000 BCE, or before Common Era. This is the first time I can find a mention of dementia in the literature. Ancient Egyptians talked about the forgetfulness that comes in older age, a perishing of the mind and forgetfulness in papyrus scrolls. In ancient India, there were texts that actually talked about how you use herbs to treat age-related cognitive decline and memory loss. Plato, who was one of the famous ancient Greek philosophers, talked about dementia-type symptoms, this cognitive decline and aging. So did the ancient Chinese scholars. In this one of the early books, The Yellow Emperor's Internal Classic, there are mention of dementia symptoms. More Greeks, so this is Hippocrates and Galen, at about 150 BCE, they talked about dementia symptoms, and for the first time they actually postulated that this was something to do with your head, that was a brain, a cerebral impact. And then we had the Middle Ages. Now, not a lot of science happened in the Middle Ages, frankly. They had bigger fish to fry. People were dying from the Black Death. There was one philosopher called Roger Bacon who talked about senility, or sort of like dementia symptoms, and he attributed this to original sin. So not a lot of progress in that phase. But in the early 1800s is when modern dementia research really kicked off. So these two guys, this is Pinel and Esquirol, they are French philosophers, psychiatrists, early psychiatrists, and they defined dementia as we do today, which is a change from your normal cognitive function, this steady decline in memory, thinking, planning, reasoning, that eventually takes over all of your brain's abilities and causes you to be bedridden. In the 1870s is about when we had some technological advances that made it possible to start going into the brain and understanding what's changing, what's actually causing these symptoms. These advances were things like microscopy. I'm showing you an early compound microscope here, but also fixation, aldehyde fixation of brain tissue so it didn't rot, so you could look at it. Silver stain, so ways of making cells in the brain visible. And with all of these advanced technologies, we started to have people looking inside the brains of people who lived and died with dementia. And the pioneers of this I'm showing you up here. So I've got Oscar Fischer. He described in 1907 in a paper something that he named plaques in the brains of people who died with dementia. And for a long time, they were called Fischer plaques. Not anymore, but they're still called plaques, so try and keep that in your brain. The other two people I'm showing you are Alois Alzheimer. So clearly, the most common cause of dementia, Alzheimer's disease, is named after him. And his trainee, Solomon Carter Fuller, who was the first black psychiatrist in the United States, he's actually descended from slaves. Alzheimer and Fuller looked in the brains of people with dementia, and they described two pathologies, plaques, the Fischer plaques, but also tangles, neurofibrillary tangles. We're going to come back to these as well. So try and remember, plaques and tangles. So they described what was going on in the brains of people living with dementia, but it took until about the 1980s, 70s, 80s, 90s, with the molecular revolution for us to understand what those plaques and tangles are made of, and to start to get an idea of how they might be damaging the brain. And it was only in 2023 and 2024 that the first treatments that can actually slow progression of dementia were approved. So that's 4,000 years. Why did it take us this long? We'll talk a little bit about that. So before we delve into how the brain changes in dementia, we need to understand a little bit about the healthy brain. So I'm going to give you a Neuroscience 101 lecture, very briefly. So you'll all be aware of the brain, where it is in your head, but I don't know if you quite appreciate how amazing your brain is. And I'm going to start by showing you, this is just a cartoon, of course, in the purple, but I'm going to start by showing you a postmortem section through the brain at the level I'm showing you here. So this is a coronal section. And it's beautiful. So you've got gray matter going around the outside in the cortex, and that's where the cell bodies mostly live. The white matter is where those cell bodies of neurons send the wires to talk to each other. And I'm going to point out this beautiful curly bit down here, shaped like a seahorse, called the hippocampus. That bit of the brain is absolutely essential for forming new memories. If we zoom in a little bit, so you all have a pretty good idea of the scale of the brain because you know the size of your own head. So you can see the scale there is 3 centimeters. If we zoom in, like those original pioneers did, to look inside the brain, now I'm showing you drawings from Santiago Ramón y Cajal, who was one of the pioneers of neuroscience. He used those really cutting-edge microscopy and silver staining and fixation techniques that I told you about from the 1870s. In the early 1900s, he drew brain cells. And the ones that I'm showing you that he drew here are neurons. The neurons are the cells that make the network. They're the ones that fire, that talk to each other, and they're absolutely beautiful. So you can see that from Cajal's drawing here. So you can see the cell body. That's where the DNA lives. That's where the programming goes. But you can also see they have these beautiful tree-like processes, and on those dendrites, those tree-like processes, you can see little leaf-looking or little thorn-looking protuberances. Those are called dendritic spines, and those are where the neurons talk to each other. Just for scale, that's a human hair at the same scale, so they're tiny, right? And I'm going to take you a little bit tinier, so inside the little red box there, we're about to zoom in to how the neurons actually talk to each other in synapses. So this is my favorite part of the brain. This is a synapse, an electron micrograph. And what I'm showing you here is a presynaptic side. It's colored in pink. That's where the neuron signal coming from. So when a neuron fires, so there's an electrical signal in the cell body, it travels down the long axon process. It's an electrical signal. And when it gets to a synapse, it causes the release of neurotransmitter from these little circles that are full of neurotransmitter in the presynaptic terminal. That electrical signal then is a chemical signal, the neurotransmitter, that's released into the synaptic cleft up there, and it's picked up by the postsynapse. Those are the little leaves, the little thorns you saw on the slide before. The neurotransmitter receptors there pick up that chemical signal and generate a new electrical signal, so propagating that signal through the brain. This is how your brain works. This is how you sense things. This is how you move. This is how you think. This is a video that might work, we'll see, that is showing you just how complex your brain is. So this is the cube of human brain, post-mortem donated brain tissue. It's 0.03 millimeters in diameter, or sort of each side. And what I want you to appreciate is just how full your brain is. The drawing I showed you from Cajal just showed 2 cells, right, or 3 cells. But your brain, the gray matter of your brain, which is what this is absolutely packed. You can see, if you're really eagle-eyed, little synapses flashing by as we zoom through this 3-dimensional reconstruction. You can see some of those axons, they're coated in fat. You can see them running through the dendrites. This is just to give you the idea of how complicated your brain is. I told you about the synapses, right? So in your brain, there are approximately 100 trillion synapses. To give you a sense of the scale of that, that's more synaptic connections in your brain than there are stars in our Milky Way galaxy. Right, so this is coming back to that 4,000-year problem. Why has it taken us 4,000 years to start to treat these diseases? Your brain is phenomenally complex. We don't fully understand how it works in the first place, so curing these diseases, of course, is quite a challenge for us. But we're getting there. So now I'm going to take you into the Alzheimer's brain. So dementia is the symptoms that I'm sure you're all pretty familiar with, unfortunately. Dementia is an umbrella term, and those symptoms can be caused by lots of different pathologies. And we're going to focus in on Alzheimer's disease today, which is the most common cause of dementia. About 60% of people with dementia have Alzheimer's disease. So I'm just reminding you here, this is the healthy brain, right? Coronal sections are here. I'm going to put one up of someone who died with Alzheimer's disease. And even though you're not neuroscientists or neuropathologists, you can probably appreciate that that one is different, right? You have this massive shrinkage. That's the brain dying over time in Alzheimer's disease. I remember I pointed out the beautiful curly hippocampus. That's here. That's almost gone. And that's why memory is one of the first symptoms, memory loss, because that part of the brain is particularly vulnerable to disease. And if we zoom in again, on inside the brain tissue, we're going to go into the gray matter. These are the plaques and tangles that Fuller, Alzheimer, and Fisher described. Plaque on the bottom and tangle on the top. This is the silver stain, the same kind they used in the early 1900s. But since then, in about the 1980s and 90s, we were able to understand as scientists what these are made of. So I'm going to show you that up here. So the plaques are made of a really sticky protein called amyloid beta. So amyloid plaques. It's going to be important, so keep it in your head. And the tangles, the neurofibrillary tangles, are made of a sticky protein called tau. So amyloid plaques, tau tangles. And amyloid and tau are both proteins that are found in all of our brains, But during the disease process, they aberrantly clump, become sticky. It's kind of like if you took an egg, you whisk it up, it's liquid, you put it in the pan, and it goes pretty gelled solid. These are solid aggregates in your brain. The plaques are outside the cell bodies, and the tangles are actually inside the neurons. And both of these are thought to be quite toxic, especially the tangles. So I told you that there are a couple of treatments, right? So there are 2 new treatments, and both of them attack the plaques, amyloid plaques. And what the treatments are actually antibodies. You can't see the bottom one. They're called lecanemab and donanemab. These are antibodies that are injected into the blood vessels. They get into the brain, and they clear this amyloid, which is pretty remarkable. So you can have a brain full of amyloid, you get these antibodies injected into your blood vessels, and they clear the amyloid. And what that does in people who are in the early stages of Alzheimer's disease is it slows the progression of the disease by about 30 to 40%. What that equates to is still debated, but it's something like 6 months of maintained cognition before you progress to the next stage of Alzheimer's. So it's not a huge benefit, but it is removing one of these pathologies and significantly slowing cognitive decline. However, they're far from perfect because these treatments are dangerous. They come with serious side effects. Some people have brain bleeding. Some people have brain swelling. They cost an absolute fortune to deliver. You have to have an injection every 2 weeks and a brain scan every few weeks to monitor for these dangerous side effects. And because of that, here in the UK, the National Health Service has not approved these, so that we can't get these for free. They're not paid for by our National Health Service. They have been approved here to be prescribed privately, but you can imagine how that's increasing health inequities around the world. Some parts of the world, these drugs are approved, some aren't, and there are good reasons for this. They are expensive, they're risky, and they have a very modest benefit. So what are we doing about this? So in my lab at the University of Edinburgh, we're trying to really better understand how the brain changes in order to develop much more effective and safer treatments. I'm taking you into the lab here. Hopefully you can see my hands in purple gloves. And one of the ways that we do this is we look at post-mortem donated human brain tissue, which is an amazing resource. We run it back to the lab, and you can appreciate they're quite small samples, so you'll see the little bit of gray matter there. And we bring them to the lab, we chop them up into even smaller samples, and this, of course, is only if somebody agrees to donate their brain and their next of kin provides consent, since they're such a gift for us. We fix them in the same kind of aldehyde fixation that was used in the 1870s. Then we put them into, we dehydrate them with alcohol, we put them into a plastic resin. You're going to see me dropping these tiny bits of brain, so you can appreciate they're very, very small, into these little pill capsules, covering them in a resin, and when we bake this resin, it becomes a hard plastic. So you see me putting them in the oven there. Why are we baking brain? Well, because synapses are so small that you can't look at the proteins inside of them with traditional microscopy. When we bake this resin into a hard plastic, we're able to use a diamond, the diamond knife at the end of the blue boat, fill in the blue boat with water here, and we're able to section the brain tissue so it's thinner than an individual synaptic connection, and that lets us look inside them. Why are we looking inside synapses? I told you about plaques and tangles, hopefully you still remember those, right? But of all the things we can measure in the brain, the thing that tracks most closely with symptoms of Alzheimer's disease is loss of these tiny synaptic connections. So we're trying to look inside them. So now we've got a long, very thin sections, but lots of them, so they're serial sections, building up that 3D picture that I showed you earlier. And we're using immunohistochemistry, so we're staining this brain tissue for things we want to look at, which is synapses, the amyloid, stuff in plaques, and tau, the stuff in the tangles. That's Rosie Jackson. She actually did her undergraduate degree here at St. Andrews, and when she was doing her PhD, she made this lovely video for us. So here's an example of the kind of data we get from the process that I just showed you. I'll talk you through it, it's a bit complicated, but it's also beautiful, so bear with me. So the yellow stuff, we've tagged the amyloid in yellow with a fluorescent tag. So you can see a big plaque up there at the top. We've tagged post-synapses, so remember that's the side that receives the signal, the little leaf or thorn. We've tagged those, and they're colored cyan in this image. And if you look over here, zooming in on a single postsynaptic structure, you can see there's yellow in it. And what that means is amyloid, the plaque protein, clumps up inside individual synaptic connections in the brain, where it's associated with synapse loss. So one of the things that Fisher and Alzheimer and Fuller discovered, the plaques, is causing some of the damage and some of the symptoms by directly damaging the synaptic connections, which are the things that let you think, that let you learn and remember. Similarly, we looked at tau. So remember we had amyloid plaques, tau tangles. So now I'm showing you a tangle. Again, the tau is in yellow here, and there's a beautiful tangle that's inside the neuron, a big thing. And in this picture, we've also colored the presynaptic side in magenta, so that's the side that's sending the signal, and the postsynaptic side is still in cyan. So when you see a little snowman-like structure, let's see if I can find one here, you can see these two little dots next to each other. That's one of those synapses that I showed you with electron microscopy. And then we're able to zoom in and say, what's inside these individual synapses? And we can, oh, it's on this side this time, sorry. We can occasionally find individual synaptic connections in the brain that contain tau. So again, these tangles that were first described in 1907, one of the things that they're doing to damage the brain is damaging the synapses and causing them to die. But the other big puzzle for tau is how is it spreading. So what I'm showing you here is a cartoon of the brain, and the point of it is to say that during the progression of Alzheimer's disease, the tau starts in the temporal lobe, and then it spreads out to that cortex that I was showing you. It spreads around the brain. And everywhere tau goes, synapses die, and cells die, and that's what's causing that brain shrinkage. So we want to know how is tau damaging, how is tau jumping through the brain, so that we can try and stop it. And one of the ways it's jumping is through synapses. Now this is a little bit of data from mice, where we injected human tau in a part of the brain that you can see over there, and then we can see it jump to the hippocampus, the part of the brain important for learning, and it jumps through synapses. But this is in a mouse model system. And lots of people around the world have shown this, but we wanted to look at whether this could be happening in people. So in human brain, again using the same technique I was showing you, we can see tau in the presynaptic side, that's the side over here, and there's an example of that at the top. We can see tau in both sides of the synapse, and that's the example in the middle, and sometimes we see tau only in the postsynaptic side. And what it looks like is tau is jumping through the brain in humans by going through these synaptic connections. So we're trying to stop that now with our current research. We're trying to figure out how to stop tau from spreading, and we think that's going to stop the disease in its tracks. But this is research going on in a wet lab now. You saw me with the gloves, and that's going to take a while. So what can you do now while we're working on these new treatments? So there are three things that I would encourage you all to do. You can look after your own brain. You can reduce your risk of developing dementias. You can participate in research if you're willing and able, and you can advocate for us. So to protect your own brain, there are lots of things you can do. You can exercise, you can eat well, you can keep socially active, physically and mentally active. Those are all good for you. Avoid things like smoking, head injury, being overweight, and being sedentary. In terms of participating in research, here in the UK, you can go to joindementiaresearch.org, see if there are any studies available for you to participate in, either as someone who doesn't have dementia or someone living with dementia. And finally, you can advocate for us. You can tell people, you can vote for people who will fund research. So, I'm going to leave you with these thoughts. First, your brain is amazing. Second, dementia is a big problem, but we will solve it with science. And third, there are things you can do now to help us in the fight to defeat dementia. Thank you.